Hello, my name is Sophie Kwan, and in this conference, I would like to discuss about gene editing, especially GMO and CRISPR. So, before I start my presentation, these are the articles that I use for this presentation, and I would like to start with comparing CRISPR and GMO. So, what is CRISPR? CRISPR technology is one of the evidence that gene editing produces a new gene therapy. CRISPR is a simple tool for editing genomes, so it can alter or delete DNA and therefore it can cause of evolution as well. This technology can be applied to various areas such as animals, humans' welfare, and therapy as well. Compared to GMO, it has larger area, including human health as well. However, on the other hand, there is another gene editing technology, which is GMO. GMO means genetically modified organism, which means that an organism's gene or genes are artificially modified for a specific reason. The reasons are usually for humans' well-being. It includes a plants, animals, microorganism, or other organism whose genetic makeup has been modified in laboratory using genetic engineering or transgenic technology. It can create new traits because it produces various combinations of plants or animals. In order to explain the mechanism of CRISPR, I will use one of the typical example of CRISPR, which is CRISPR-Cas9 that is related to COVID-19 vaccine. So firstly, DNA invasion must be occur. In this stage, foreign DNA from a virus or plasmid invades the cells, so the invading DNA can be incorporated into the CRISPR array. DNA fragments from the invading DNA are incorporated into the CRISPR locus as spacers. Then, pre-cRNA transcription happens. The cell constitutively transcribes a repeat and spacer group into pre-cRNA. After this, guide RNA, which is called gRNA, is formed by constitutively expressing transactivating RNA base pairs with the CRISPR repeat sequences on the pre-cRNA. Due to the formation of gRNA, Cas9 protein binds to the gRNA and becomes activated. The activated guide RNA complex binds with the target DNA. Then the Cas9 protein cleaves the invading DNA and inactivates. With this method, CRISPR can be applied to various areas, such as pathogen diagnostics, corrections of deleterious mutation in embryos, genome editing for improved animal breeding, gene drives to eradicate malaria, and cell therapy. RT-PCR is one of the example of CRISPR-based tool, and it is for diagnosis of COVID-19. So the process starts with reverse transcription by isolating the viral RNA, and then PCR happens. And as you know, PCR has three steps: denaturation, annealing, and elongation. And then it detects whether the patient has the disease or not. On the other hand, I will explain the mechanism and applications of GMO. So for the GMO mechanism, it firstly identifies a trait of interest by screening the list of chosen plants. Then it isolates the genetic trait of interest. At this stage, comparative analysis of genomes will be used to identify traits. The next stage is an actual gene editing step, which is inserting the desired trait into the new genome. Genetic trait is cut and pasted into a plasmid using enzymes. After that, controlled growth chambers are used for growing the GMO monitor to ensure that the new GMP grows and replicates. For the application of GMO, it has relatively a limited range of area compared to CRISPR because it can be only applied for plants and only for few animals. So the first example is soybean that has herbicide tolerance. It was conferred by expression of glyphosate tolerance. And the second example is corn that has insect resistance. Researchers and farmers modified the corn to protect their productions from the pest. And the next example is canola that has different fatty acid composition. This specific canola has a high LARTS level achieved by inserting gene for ACP, which is California bay tree. And the last one is plum that has virus resistance. It has a resistance to pox virus, and this could be possible by inserting the code of protein gene from the virus. Even though the gene editing has various advantages that I mentioned previously, there are some people who are against gene editing, and they claim that it is very dangerous to use as a technology because it can be transferred to the next generation, and there's still unknown and unexpected side effects, and it involves human embryos, which means that it is contradicting with moral and religious part. And specifically, people concerned about enhancements or abuse of gene editing, so they are against for gene editing. However, on the other side, people claim that the gene editing technology is beneficial and necessary for the future and the current situation as well because it is one of the useful technology that can produce as therapeutic usages. Also, International Summit agrees that germline genome editings are allowed. They think that it doesn't have any problem to use as a technology. This argument is still ongoing process, therefore the entire world and scientific organization must be working to satisfy both sides' arguments. Concern. Through this conference, I learned a lot of things even though the concept was already familiar with me. Because I take IB chemistry and biology class, the content was familiar with me. However, I was able to have a deeper understanding by researching a specific example and its mechanisms. It was really great opportunity to participate in this conference, and I hope everyone enjoyed my presentation.